Dear students, today I will talk about the histological structure of the kidney. If you have a first look at the hematoxylin stained slides of the kidney, this is really, it seems to be chaotic. You can see almost randomly distributed cells. Hopefully, by the end of this lecture, everything will be much more known for you. Whatever you can recognize easily is a thin layer of dense collagenous connective tissue on the top of the kidney. This is the kidney capsule. This is what we name the renal capsule on the anatomical part of this structure. This is the capsule of tunica fibrosa. If we have a closer look at this tissue mass, you can see we have two types of tissue. Some of them round. This can be capillary tufts or cross-section of tube. This is what we name the cortical type of tissue. This cortical type of tissue, as I talk about in the anatomy lecture, is located in the cortex cortices, in the renal column, and in a part of the stria medullaris cortices. You know, here we have the renal lobule and the external parts of the renal lobule, which is what we name the pars convoluta lobuli cortices, also contains cortical type of tissue. The other type of tissue contains elongated structure, and this is what we name the medullary tissue because it's predominantly located in the medulla of the kidney. These are located in the middle part of the renal lobule. This is what we name the radiate part of the lobules, pars radiata lobuli cortices, and also the entire medulla is filled with that. You can see thicker and thinner but elongated tube. Here's the cross section of the medulla. After this introduction, let's see what really happens in the kidney. Kidney, as a parenchymal organ, it has a large number of fully functional units, and the fully functional unit of the kidney is named the nephron. We have a large number, between 1 and 2 million nephrons in a kidney, and they make total of 80 kilometer canals together. In the microscopic slide, you cannot distinguish the nephrons. You cannot differentiate from nephron to the other, so you cannot tell that this particular tube belongs to this or any other nephrons. So you are not requested to show a nephron. However, you have to know the nephron in details, and you have to identify that which part of a nephron is this, this, and this, and this. Now let's see what the nephron consists of. The central component of the nephron is the so-called Malpighian or renal corpuscle. This is a round substance, and the primary function is to make the filtrate. This is about 0.2 millimeter in diameter, just in the borderline of the visibility. And in the middle, we have a capillary tuft, which we named glomerulus. Be careful not to mix the entire Malpighian corpuscle with the glomerulus. Glomerulus is just a part of it. In the glomerulus, we have an artery which is coming in. This is the afferent artery. And the efferent artery, interestingly enough, the blood which goes out of the Malpighian corpuscle is still in high pressure, and the vessel is also another artery. Between them, we have the capillary tuft and mesangial cells. The entire Malpighian corpuscle is surrounded by a flat epithelial tissue. This is now the Bowman's capsule. Here in the realistic picture, you can see these flat, elongated nuclei. This is the parietal part of the parietal lamina of the Bowman's capsule. This is continuous to the surface of the vessel. These violet color cells, violet labeled cells, are the visceral lamina, which is covering the surface of the capillary tuft. Between the parietal and the visceral lamina, we have a cavity, and this is where the filtrate is located. The Malpighian corpuscle has two poles. One of the poles in which the artery is entering and leaving, this is named the vascular pole, and just opposite to that we have the first tube which carry out the filtrate, and this is what we named the urinary pole. Okay, from the Malpighian corpuscle, tubes come out, and we have three types of active tubes there. The first one is named the proximal tubule, which is immediately connected to the Malpighian corpuscle. Proximal tubule of nephron is about 10 to 12 millimeter long. It's at the level of histology is quite long, and 40 to 60 micrometer in diameter. Typically, it has two things. They have a brush border. You will see later on in the ultramicroscopic slide what it looks like, and basal stripe. So these two should be remembered. If you see a light microscopic picture of it, you can see that the upper surface of these cells, these are typical light microscopic picture of proximal tubules, is washed out. You cannot see a clear upper surface of the cell because it has large number of microvilli, and this is washes over the entire picture. This proximal tubule consists of two parts, one of them coiled up nearby the Malpighian corpuscle. This is what we named the convoluted part. After that, it turns straight and goes down in the direction of the medulla, and this is the straight part. Here I'd like to call your attention that in the different book, you came across different terminology. The very old books and the modern books that we turn back, we name this tube is the straight part of the proximal tubule. Some books, about 10 to 20 years ago, they considered that everything which is straight is the Henle's loop, and we have thick and thin segment of the Henle's loop. Nowadays, it's worldwide agreed that the Henle's loop only the thin segment, and this segment supposed to be or considered to be part of the proximal or later the distal tubule. The Henle loop, the real Henle loop, this thin segment, consists of a simple squamous epitel, and this is about 15 to 13 micrometer in diameter, and it has a relatively thin 1 to 5 micrometer wall. Further details later on. The distal tube, which is a continuous of the Henle loop, is about the same size, slightly longer, can be slightly longer than the proximal tube, 10 to 14 micrometer, and approximately similar size in diameter. The cells have even, the tube has even lumen because the cells have no microvilli on the surface, but they still have the so-called basal stripe. Similarly to the proximal tube, this also has a straight part which going up to its own Malpighian corpuscle, so it will be faithful for the origin, and we have a convoluted part which turns around. There is a very interesting and medically very important piece of the distal, and always the distal tubule. A certain segment of the distal tubule become in contact with the Malpighian corpuscle just at the vascular pole, and those cells which are in contact are modified. Here you can see the realistic picture. This is the area that we're talking about, and this is what we made the macula densa. This is one of the key components of our most powerful blood pressure control system. You will see later on details. Finally, we have a short collecting tube. This is already not active. It's already the ready-made urine is there, and this is connects to the ready-made urine to the papillary ducts, the ducts of the medulla papilla. Okay, let's see some further details. So now, hopefully, you become a little bit more familiar with this system. Here you can see Malpighian corpuscles, which is predominantly capillary tufts. You can see capillaries and related cells. And here you can see around the Bowman's capsule. Around the Malpighian corpuscles, you can see tubes. These are the proximal tubules. This is a little bit more dark and is a very washed out, not really clear borderline of the cells. And we have distal tubules, which are somewhat paler, and more importantly, they have very nice even lumens. You can see a certain line of the surface of the cells. This is a kind of cortical type of tissue, once again, where we have the Malpighian corpuscles, the proximal and the distal tubules. This part of the picture contains medullary type of tissue, also elongated tubules, but also two types. this is proximal. You can see washed out the surface darker cells, and distal tubules, much more clear surface and somewhat paler cells. The cortex, so once again we have a Malpighian corpuscle and the convoluted tubules. The medulla, we have the straight tubules, and at the end, when we close down, these tubules become bigger and bigger. The cuboidal epithelia turns to be columnar, and at the papilla will open into the lesser calyx. This area is named the area cribrosa. The nephrons, all of the nephrons looks basically the way I described, but there are slight differences between them. Those nephrons, which are very close to the cortex, what we name the cortical nephrons, these are oh, well, it's supposed to be something. Okay, cortical nephrons. These are slightly different. These are located in the cortex, cortices, and especially the juxtamedullary nephrons. These ones. These are close to the medulla. This is the in the renal columns and the stria medullaris. They have a slightly different feature. Whatever I described, I show the proportion is valid to juxtamedullary nephrons, in which we have a very long Henle's loop, unlike the cortical nephrons, which has a short Henle's loop. Also, the distal tubule has a different proportion. We have a much longer distal tubule in the cortical type of nephrons and shorter in the juxtamedullary nephrons. Also, the vessels are different, and that I will describe later on with the vessels. What about the blood supply of the kidney? As you remember from anatomy, the arteries, the branch of the renal artery comes in, and in the sinuses is divided the radiator lining arteries, which aiming at the surface between the renal lobes, and this is what we named the interlobal arteries. The interlobal arteries at the borderline between the medulla and the cortex, connected by an arch-like arteries. These are the arcuate arteries. And from the arcuate arteries, short arteries going straight up to the cortex, and these are the interlobular arteries. You can see enlarged in this picture. These interlobular arteries behave a slightly different way. We have the superficially in the caudal cortex, in which we have star-like branches of the arteries. This is what we name the stellate arteries. It's not connected to Malpighian corpuscles, just supply the surface. In the cortical area, the upper area, most of this area, the branches of the interlobular arteries are the afferent arteries, which carrying the blood into the Malpighian corpuscles, into the glomerulus part of the Malpighian corpuscles, and the outgoing artery still has high pressure, are the vas efferent. This is continuous in the so-called cortical peritubular network. It means that the vas efferents capillarize again, and it's going around the tubes, proximal and distal tubes of its own tube, whatever is called the part of the particular nephron. This is, as you notice, a double capillarization. First, the glomerulus between the vas efferent and efferent, and we have a second capillarization of the same artery around the tubules. One of the important things to remember is that in the glomerulus, we still have high pressure blood. Even a vas efferent, we have much higher than the normal venous blood. This is required for the filtration. You know from biophysics, if with the physical pressure you act on a filtrating liquid, it filters quicker. This is what we name the ultrafiltration. So this is similar to the portal circulation, but difference is that this is not a vein, but this is the artery, the intermediate vessel. In the medullary part, we have straight long arteries going down to the medulla. This is the arteries in the area of the juxtamedullary nephrons. and this differs. Some of them just going down directly from the interlobular artery, sometimes from the arcuate artery, and supply the tubes in the medulla. This is forms 10% of these arteries, and this is named the arteria recta, straight arteries. We distinguish real straight arteries, arteria recta vera, which coming directly from that. And we have the second one, the arteria recta spuria, the false arteria recta, which is practically the vas efferents of the extramedullary nephrons. Both of them, once again, supply the medullary tubes. Extramedullary nephrons vas efferents is that one. Some people think that the arteria recta vera, which is not related to Malpighian corpuscle, is a kind of consequence of damaging the Malpighian corpuscle, but has no practical significance. So the medullary peritubular network is fed by the arteria recta system. The veins, very similar to the arteries, from all of these branches of the interlobular artery, the blood finally goes to the interlobular veins, which goes to the arcuate veins, the interlobal veins, and then through the interlobular vein to the renal vein. Lymphatic drainage. Practically every organ has lymphatic drainage. Maybe the brain is the only exception, and the kidney also does have. The most lymph is produced around the Malpighian corpuscle. So in the periglomerular space, the majority of the lymph is produced, and the lymphatic vessel will conduct this lymph all the way till the lymph nodes. As you know, that the lymphatic vessels always run parallel with the nearby veins. A practical suggestion: if on the exam you ask the lymphatic drainage of something, and if you have had little memories on that, but you remember the veins, over 90% you figure out that if you say this way goes this and this way, where the veins of this particular organ go, and the lymphatic vessels, if they do have name, also named after the similar name as the veins. This happens also, so parallel the interlobular arcuate and interlobal veins, the lymphatic vessels go, they step out through the hilus, and they will end up in a nearby lymphatic lymph nodes, which is named the para-aortic lymph nodes. The kidney also has nerves. These nerves are autonomic nerves and belong to the sympathetic system. Nearby we have the sympathetic trunk with the chain of ganglion. Approximately the second lumbal ganglion is slightly bigger than the rest of them one, and it is named the aortico-renal ganglion. This ganglion sends postganglionic fibers in the adventitious of the artery. You remember the primary task of the sympathetic postganglionic fibers is to control the blood, controls the smooth muscles of the arteries, consequently the blood supply of the organ to which the artery carries the blood. Even although it controls the arteries of the kidney, but this is not of life importance. So if you transplant the kidney, we cut a nerve, and it's not regenerate, the kidney is functioning pretty well. Moreover, we might have a problem with that. If you're undergoing strong stress, then under this distress, the arteries of the kidney can shrink, and the blood supply of the kidney can be reduced beyond the tolerable level. Kidney has works with needs a lot of energy, and it's very sensitive to oxygen and nutrition. Consequently, for a short period of time, it has reduced that. It has certain quantities of the kidney tissue is damaged. This is what we name this stress kidney, with the consequences you study later on during your clinical studies. Now let me make a kind of functional summary. You hear a lot of very nice important information, important pieces of information, so also details on the function of the kidney, but maybe you are lost between the lot of information. Let me briefly summarize. What does the kidney do? How it carries out its product? First of all, the first step is the filtration. So from the large quantities of blood passing through the kidney, 10% is filtered out. You will see later on the most important component of the Malpighian corpuscle is a basement membrane. This is the filter, which let all the molecules passing through, which is lower than about approximately 20 kilodalton. This is an important piece of data. So anything smaller than that, it will appear in the filtrate. Whatever is bigger than that, like the blood protein, will all remain in the blood. Then this passes through the tubules, and getting back, 99% will be reabsorbed, and only at the end of this tube system, altogether 1.5 liter per day urine stays. Why we have these things? The primary part of one of the most important function of the kidney is to get rid of the unwanted materials. We can say poisons or toxins or any kind of garbage which appears in the blood. How can you do that? There are two theoretical possibilities. If the kidney is very clever enough and they even know that what type of chemicals will the food industry poison us in next 1,000 years, they could make a mechanism that it will get out from the blood all of these toxins, and we clean it. Practically, it's impossible to do that. It does the different way. It discharges everything, which is of low molecular way, and takes back whatever it knows, whatever is necessary for us, and leaves in the garbage, in the urine, whatever you don't know what is that, or approximately we don't need that. It is very similar if you want to clean your apartment. One of the possibilities, an easier way, the first way is, whatever you see, it's not, you don't want to, put in the garbage can and dispose it. However, this way, a lot of things remains in your apartment, whatever you don't need or whatever is unnecessary for you, but you don't mind. The other way you're cleaning the apartment is push everything to the street and get back what you want and leave there what you don't want. This is very tiring, but this is absolutely useful because just what you really need will get back. This is the second type of cleaning solution is done by the kidney. So it pushes out everything and gets back whatever is good, whatever we want to, and leaves in the urine whatever you don't know. This way, doesn't matter what type of novel things we are poisoned with, it will be in the urine. How it's done? The proximal tubules absorb this sodium. The water, even although we have some kind of channels like aquaporins and so on, they are not really efficiently we can mobilize the water. The water always goes passively physically after the osmotic pressure. So the first thing, the largest quantity of ions which gets into the filtrate is the sodium. The primary function of the proximal tubules is get back the sodium, 99% of the sodium, whatever was in the filtrate. This is a very energy demanding way, but we can do that with energy because the sodium has an ion and it can be moved with the appropriate mechanism. This is so in the proximal tubule, especially the sodium gets back, some other things also, but this is the most important thing. What will be the consequence of that? In this, the filtrate will be more and more diluted because the sodium getting out, and in the blood vessel around the proximal tubule will be more and more concentrated sodium solution, so there will be a big osmotic difference between the inside and the outside. Then everything goes to the Henle's loop, where we have very thin cells. Apparently, they do not have a lot of energy, can do actively things, and this is their real task. Whenever we have a very diluted water inside, and a very concentrated water outside, simple, because the water knows the physics, will go out in the direction of the osmotic concentration, and this is where passively, without significant energy, all the water follows the sodium in there. But at the end of the Henle's loop, approximately most of the necessary water and the sodium absorbed. Now it comes the distal tube, and this is the clever tube of the kidney. This is practically whatever we can control. The proximal tube is like the gorillas of the Godfather. They are strong but stupid guys. They do only one job, and they do it very well. And the distal tube makes a kind of fine adjustment. So this also energy demanding way. And they exchanged sodium to potassium, potassium, hydrogen. It will absorb or not absorb phosphate. Most of the sugar also comes there. Sugar also comes for the other tubes, but this is also. So this is what makes the kind of fine adjustment of the ion concentration, and this reabsorbs whatever is still needed in a relatively higher quantity. So everything which is controlling the modifying the function of the kidney acts on the distal tubule. And finally, it will be the collecting tubule and so on. So this is not a complete description, but I hope you get a basic idea what the kidney does, and a lot of interesting details you will study with other subjects. Now, up to this point, you have the general idea on the histological structure of the kidney. Now, let's a little bit refine. Let's see the fine structure of the nephrons. Okay, in this case, we get a detailed description of all of the components I talked about before in the previous part of this lecture. You can see a scanning, first of all, a scanning electron microscopic picture on the glomerulus. You can see this very nice capillary tuft. This is a realistic picture. This is light microscopic picture with the Bowman's capsule and the glomerulus. Let's talk about the Bowman capsule. As I mentioned, we have a Bowman's capsule has a parietal lamina. The parietal lamina is simple squamous epithel, and as you can see, in the electron microscopic picture, it's not as thin as the endothel, but still a simple squamous epithel. This is the function of them is to seal this area and prevent the filtrate to flowing away and directly to the proximal tubule. The visceral lamina, at the vascular pole, it turns to be continuous to be the visceral lamina. In the embryology, in the development of kidney, you will understand why and how. And this visceral lamina, cells of them will be modified a very interesting way, and they are cells with a long processes and many fingers. And this is what we named podocyte. You know, podos is Greek for food. Podocytes is cells with food. You can see beautiful podocyte here with primary, secondary, and tertiary legs and fingers going away. So this makes the visceral layer. Further details a little bit later on. Between the parietal and the visceral layer, we have the so-called periporal cells. It's labeled green in this picture. Interestingly enough, they have vesicles inside the cytoplasm. Very probably they produce some kind of control substances, but up to this point we don't know what. It may be the task one of you to discover what is the function of these periporal cells. To continue the fine structure of the Malpighian corpuscle, let's talk about the glomerular endothel. The total surface of the glomerular endothel is about 1.5 meters. This is pretty much fits the information whenever the people undergo dialysis, the artificial kidney. The surface of this instrument is also very similar. The endothel is fenestrated. Fenestra, you know, it's Latin for window. It means little windows on that. It's around 1/10 of a micrometer, and in the contrary of that, the cells, the endothel cells, is sealed to each other with tight junction. Here you can see a cross-section of this area. This is the endothel, and these openings are the windows. And this is the endothel's cytoplasm. Here you can see also another electron microscopic reconstruction picture. You can see these openings are the fenestrae. Some of them are covered with diaphragm. Some books think this is interesting. I think it's just a kind of problem, a kind of developmental problem which does not have any practical significance. Outside of the endothel, we have the basement membrane, and practically this is the most important component of the Malpighian corpuscle because this is the filtration surface. All the other cells present there is just surfacing these cells. The endothel prevents the blood clotting, just to isolate the blood platelets and the erythrocytes to come through it, and the rest of the cells taking care of it, as we will see later on. Outside of the basement membrane, we have the podocytes, and it's a little bit disputed, but very probably the podocytes making the basement membrane and making new basement membrane if they damaged. As you will see later on, similar to the coffee filter or whatever, the basement membrane can be plugged, and there is no way to clean it other than just these cells, the mesangial cells chewing it up, and the cells, probably podocytes, making a new basement membrane. The basement membrane is much thicker than the usual. You know, the normal basement membrane is about 80 nanometer in thickness. This is much thicker, 450 nanometer. And this looks like a felt with a lot of molecules, and this is a kind of filtrating surface. They have about 2-3 nanometer pores altogether, and as I mentioned, 20 kilodalton. This is a magic number of the kidney. This is what it lets pass through. The rest of them cannot through. This has 3 laminas. We have the external rear lamina rara externa, which made of laminin. It's a kind of glycoprotein. You know what glycoprotein is, and it's very acidic, has a lot of sulfate group. So this charge probably also modifies which molecule can pass through and cannot pass through. The other component of the lamina rara is the so-called core protein. This interestingly looks like that, and this belongs to the glycoproteins, which is a sticky substance that makes a kind of integrity in the membrane. This is the type enactin or fibronectin type, very similar to these. The lamina densa, the middle layer, is a most thickest structure. They are made of collagen, type 4 collagen, and 4 units are like a hairpin is connected to each other. The internal lamina rara is very similar to the external one. Podocytes. Podocytes are the cells which is outside of the capillaries. It also can be considered as the visceral lamina of the Bowman's capsule. You can see the podocytes have many feet, primary, secondary, tertiary fingers, and this reconstruction with 2 different colors I labeled the 2 neighborhood podocytes. This is one, this is the finger of the other side, so it's very nicely alternating each other, something like this. Between the podocytes, we have a very thin membrane. This is called glomerular slit membrane, and this is the last level of the filtrating surface. This is about 5 nanometer, and this is between the fingers. This leaves alone about 2 to 3% of the area. Here you can see once again a cross-section of the wall of the capillaries. This is, as I mentioned before, the fenestrated endothel. These are very well visible in electron microscope, the 3 layers, sublayers of the basement membrane. These are the fingers of the podocytes, and this is the glomerular slit membrane. This is the last point of the filtration. Here you can see a nice electron microscopic picture of the podocyte. Here you can see this is the fenestrated endothelial basement membrane and various fingers of the podocytes. This are the neighbors, and this is the nucleus of the podocyte outside. The next is the mesangial cells. Mesangial cells are labeled green in this picture. You can see we have in two areas between the capillaries and between the arteries. Whatever is between the arteries, the ways efferent and efferent, is named the extraglomerular mesangial cells. These are part of the juxtaglomerular apparatus, and when we talk about the juxtaglomerular apparatus, I will describe in details. The other one is the intraglomerular. This is what looks like this very ugly looking guy. This is typically has uneven nucleus. The nucleus is pale and very active, and the surface is also very sophisticated. And this is a typical phagocyte. This is, if you see in light microscopic picture, many of these nuclei are nice oval. These are the nuclei of the podocytes, and some of the nucleus is pale and irregular like this and this. These are the mesangial cells, but you are supposed to recognize also in the exam. The third type of cells is very really visible, flat nucleus. This is the nucleus of the capillary endothel. The intraglomerular mesangial cells make about 15% of the glomerular mass, so quite a large quantity they located. As I mentioned, their primary task is to destroy the plugged basement membrane, which will be renewed by other cells. All the mass of the mesangial cells named the so-called glomerular matrix. And this is what I wanted to talk about you today, and we continue this fine structure of the kidney in the next lecture. Thank you very much for your attention.